again, only 4 people, and they're missing one of the most exciting lectures. Well, anyway, so today we're going to move on to chapter 6. I've graded your homework, and those are fine. I think occasional slight slip up. The exams I have not graded yet, but I will do so soon. Okay. You may have noticed that I posted an announcement that my official office hours are, well, one of them is now in the half hour before class starts, so I'm going to say till the half hour after this class to make it easier for you guys to attend. But of course, those rules are only, the office hours are only official things that we have to set in writing. You're welcome to discuss Zooming or meeting in person any other time. Okay? Any questions about anything? So today we are zooming ahead and not zooming as the video thing, but we're going on talking about chapter 6 and organization and expression of lymphocyte receptor genes, an exciting topic indeed. And note that I have the exclusions here listed, texts that you do not need to read. Of course, I hate to ever be telling students don't read something or don't pay attention to something, but things that aren't as important. Really, some of it really gets into the weeds about enzymatic detail, about cutting and pasting of genes. So this link here, which says this is stuff that you can skip, or I should say, read with paying less attention. So we'll look at this text now, and fundamentally, the major thing addressed in this chapter is one of the big questions of immunology, and I would argue even one of the big questions in biology at all, with my slant towards immunology. So here's the problem. It's good to reflect on the basics rather than just dig into an experiment. Here's the problem. A large number of potential pathogens. We know we or mice can respond to all of them. How do we handle that? All those things out there. Remember the basic dogma that we learned, basic genetics, one gene, one polypeptide chain? Then that kind of had to be modified, people say one enzyme, one gene, one enzyme, and then that kind of had to be modified for sometimes an enzyme is made up of multiple chains, sometimes different chains, but still kind of the same idea. So, but you have this need for incredible diversity that you or a mouse has to be ready for any virus, any bacterium, any fungus that might come its way or your way. How is that possible? We have a big genome, but not an infinitely big genome. Okay, so we know that. So this was really quite the question how that can be achieved. And I think you guys know the answer by now, that the book can't make it a complete mystery. They touch on it and talk about rearrangement of genes, but still we need to go over exactly the experiments that told us this answer. So the idea of gene rearrangement is first worked out for immunoglobulin genes, This was a completely novel mechanism. I can't stress that enough. This rearrangement of immunoglobulin genes occurs only in B cells. You might remember some classic experiments in basic biology and development, how they show that nuclei from fairly advanced stages of development can still allow for a complete development of a frog or some other organism, showing that the genome was not changed. That's kind of the basic things, is that the information is still there. Remember Dolly the sheep, and the idea that you can get from a differentiated adult cell, you can get a whole sheep. So that's the general rule, but of course we all love situations where the rules are broken, where there's a new rule, a new way of thinking about things. So this mechanism that we'll talk about in a minute was and is completely novel, occurs only in B cells, but not surprisingly, a similar mechanism, analogous mechanism, was found to occur in T cells to account for the generation of T cell receptor proteins. Okay, so from some early work with small molecules attached to proteins, people found that the body can recognize small differences, and people started to try to make estimates of how many antibodies needed to be made by the genome. And 10 million was thrown out. That's a lot. And that's kind of an underestimate. So people began to address this big issue. And it's important to think about how science works, that sometimes the technology kind of drives what we know. It's not like, I think we've all heard the story about Einstein and the patent office just thinking and coming up with great thoughts about physics. Well, biology isn't so much like that. It's largely driven by technology, what we can do. When we learn to be able to sequence proteins, when we learn to be able to sequence DNA, things like that, right? So at some point, people began to be able to sequence proteins, and they found that, they looked at antibodies, that the first 110 or so amino acids in antibodies were quite variable. And the next group, we now call the constant region, was not truly constant in that it had only one type, but it was clustered into a few small groups. Okay? And this kind of work, again, technology, this was done with B cell tumors. So myeloma's a tumor. What's the advantage of that? Well, we just talked about diversity, and that's really great for the immune response, but for the scientists trying to figure things out, that's a little harder, right? If all the antibodies are different and you want to do some sequencing. So they did a lot of early work with mouse or human leukemias, white blood cell tumors, and those cases, there would be a whole lot of one type of tumor, right? That's what cancers do. And people were able to sequence from different tumors, get a collection of sequences. And from that, they determined that, again, as I said a minute ago, that the first 110 or so amino acids were quite variable, and the next group clustered. And let's look at that now on this slide. Sorry that's so low. And this is our stick model of antibodies. And we now call these regions V and C, variable and constant. Realizing the constant still is a small number of sequences. And you recall, we've already had this, that here in this line diagram, we see this is like an IgG molecule, two heavy chains, two heavy or long or called heavy chains, and two short or light chains. And each of the chains has a variable region and a constant region. So this kind of data was available even from these early protein sequencing data. And it's consistent with great diversity and being able to bind to different antigens, but still some big questions here, because it goes against the rules of one gene, one polypeptide, right? How do we achieve this? There's our slide here, and found that the same constant heavy CH or CL can be connected to millions of different VH or VL, like shown here. And conversely, the same variable heavy VH region can be connected to different constant regions. So with what we know about molecular biology and the relationship between DNA and RNA and protein, how does that actually work? So some big questions here. So investigators proposed some theoretical models of antibody genetics. People tried to think of things like, well, could it be like the idea of a mold or something with an antigen and try to fit something to it? But really there was not much in what we know about molecular biology that seemed to fit that. So broadly speaking, there were a couple different types of theories. One was germline theory, and people calculated that the size of the repertoire could have to be bigger than the size of the genome, which didn't. That would not be possible. Many V's here. When I thought of variable and constant becoming the B-cell DNA being different than germline DNA. So Dreyer and Bennett suggested recombination, bringing together V and C regions. And also, there was the idea, some people suggested the idea of somatic recombination, hypermutation, with relatively few germline genes. So ultimately, we learned that both these sides were partially right. So a large but limited number of V segments is acted on by a system to create diversity. And again, this is completely novel at this time. So key experiment and credit for this result is given to a man named Suzumu Tanagawa, a Japanese man educated in the US who did this work in Basel, Switzerland at an immunology institute there that I think is closed, but a lot of great work was done there. So I was pondering myself, this is a classic experiment, and what really makes an experiment great? And I was thinking, what do we like? So it answers a question that we care about, right? A big question. How do we get this diversity? And it also gives a clean result that I think is relatively easy to interpret. The data is relatively easy to interpret, and people to say, aha, that explains this and this and this. Okay, so, and forgive me if I'm boring those of you who are familiar with this, but I think it's good to go over classic experiments like this. And the book does a great job of demonstrating the great lengths that Tonegawa and his associates did, the things they had to go through to do this experiment. And it's amazing. A lot of the things we take for granted now that we can order, they couldn't do. And this was like 1974 or something like that. So first, the book gives a lot of detail and is sort of tells the truth, but sort of fibs a little bit, and I'll tell you about that later. So that's the experiment I will describe is almost what they did, not exactly what they did. Okay? And the point here, to give you the answer, is to show that DNA in the B cell tumor was different than the germline. Germline, what it is the start that all the other cells are. Muscle cells, skin cells, all the other cells. Okay? So to do this brings up the idea of doing a genomic southern. And I think I've learned that southern transfers, southern blots or southern transfers were a commonly used technique, and I think maybe used less so in the last few years, but I think I need to go over them. Now, how many of you guys know what a southern blot is? I guess my king is right. Okay, that's correct. Okay, so a southern blot, it's interesting. You know, I love anecdotes. And so you might have wondered why some things are called northerns. You guys know what northerns are, right? Okay. The reason things are called northerns is because they were originally southerns. And southern is a guy's name. It's southern. And then when people found a new technique, they called it a northern. Because you know, molecular biologists tend to be wise asses, right? As compared to but anyway, so that's why there are northerns and westerns, because of a man named Southern. So impress your friends with that anecdote. So the southern blot and transfer allowed people to have some idea of the genomic neighborhood of a gene. So typically in a southern blot, one would take genomic DNA from whatever, human, a mouse, or a frog, and cut it with a restriction enzyme. They cut rarely, so you'd have only big fragments. I don't know if this does justice to it. Here, so we see this gel, this big. So now you have total DNA, all the DNA, something. Run it out on a gel, separated by size. This is going to give a continuous smear, right? Because you have the whole genome there. The whole frog or mouse or human genome, right? But there's a way to look at particular sequences that you're interested in. And this is described here. You, like in a northern, you apply paper to that blot, and the DNAs will be transferred to that paper in the position they were at on the gel. In other words, how big they are, how far they ran. Okay? Are we good with that? Okay. So now, so there's huge amounts of DNA here. Now, that paper with all that DNA blotted, that can be subjected to analysis by using a hybridization probe, using a particular clone that has radioactivity on it or has a color on it. So now you do this reaction where you allow this probe, we call that a hybridization probe, and allow that to occur, and then one can detect later, that's only going to detect where that particular gene is. So let's say we're looking at globin genes. You make a hybridization probe of a globin gene, run out total DNA, hybridize it to radioactive piece of DNA with globin on it, and now expose it to film. You're only going to see a signal where globin genes are, okay? Everybody get that? Tell me if there's anything that doesn't make sense. So, in my example, I believe there are 5 human globin genes. And you might see 5 different bands. It's not particularly significant how big they are, just that they're different bands. And it tells you a little bit about the neighborhood that those globin genes are at. Okay? It suggests 5 different genes. It also could be that the restriction enzyme that was used cut in the middle of one of them, so there are fewer genes, but I think you get the idea. Okay? Everybody with me so far? So this is a way you can get an idea of the genomic neighborhood, the environment of particular genes, from a mix of all DNA. You can get an idea of how many globin genes there are. So this kind of technique was done with immunoglobulin genes. That's why I'm bringing it up now. Okay? But let me know if there's anything that I'm glossing over because I'm used to thinking about this. So here we have our comparison. The Tanagawa experiment is antibody-producing tumor cells from the same strain of mouse. Same strain of mouse. Well, okay, first, germline DNA, just regular cells. It says here embryonic liver, but it could be any other type of tissue. Okay, regular cells. Here, on this side, antibody-producing tumor cells from the same strain of mouse. The same strain of mouse, so it matches up. And a tumor cell line because we want all the cells to be the same. Okay, from one, couldn't even be different tumors. We want all to be the same. So they cut DNA with a restriction enzyme and then run the gel. This is shown as a continuous smear because there's many, many fragments, right? There's a whole genome. But then the transfer occurs, and that the bands are going to transfer where they were on the gel, so we get an idea. And then this highly idealized data showed up. This was found. They used 2 different probes. Remember, a probe detects sequences like it. So these were light chain probes. And one probe detected only the constant region, and one probe detected the whole light chain. Okay? And what they found is startling. It's not especially significant, the size of the bands, but what is significant is difference between here and here. So that shows that during development, the genomic organization of this locus changed. Okay, so don't worry too much about, oh, these bands are shorter than here. But do note that that it did change between the germline and the antibody producing cells. So that is fundamentally different than anything else. This is programmed genetic rearrangement. Occasionally there's random mutation, including translocations, but this is programmed in to the system for immune antigen receptor genes. It's also noteworthy that these two bands were the same size, so suggests that both the whole light chain probe, which includes V region as well as C, and the C region were on the same band. And consistent with what we now know that they were brought together, the V and the C. Okay? Questions about the technique or or the interpretation of the results? So yes.
You were saying that they are the same length. So that is commensurate with our current knowledge that the C region and the focus on the light chain. There is a C region on the light chain.
There is a few, sorry.
There is a C region on the light chain.
The C region, there's a probe.
No, I mean so this finding that the C region and the light chain have the same molecular weight is
Yes,
commensurate.
yes, that is significant. These are on the same band. Yeah. Now, there's a little bit of chance here because I don't know if they tried other restriction enzymes. There could have been an enzyme that cut somewhere in between, but it is significant that they're on the same. And they've been brought together. What we now know is because those have been brought together.
Yeah, and that is because on the light chain there is a C region.
Yeah,
on the light chain.
On the light chain. Oh, there is a C region. Yes. That was known before.
Okay, so they sort of.
Okay.
Thank you.
Okay. So let me see here. This is actual data. So I said they didn't exactly lie to us, but they said the experiment was something like this. What they did, what Tanagawa and Associates actually did, was an even more difficult experiment, because even though that Southern technology is old for you guys, it hadn't even been developed then. So what they actually did is ran out a really long gel, and they cut slices from it. They're actually a series of blades that do that. They cut slices and then use them for a hybridization experiment. So what they actually did was even more difficult and amazing. And this is the actual data that you see here. Okay. So that's fairly impressive. So the Tanagawa experiment first shows that the V and C change in the two cell types. Later, around this time, was when people began to be able to do sequencing and be able to do larger amounts of sequencing. And later people showed that the V and C region are separated by a long distance in non-B cells. Next, they found that there was another segment which they called, in light chains, that they called a J segment, which accounted for 13 amino acids in this particular immunoglobulin. It was called J for joining. V and J are far apart in the germline, and they're made contiguous in B cells. So not only are V and C included by different segments, another segment is used. So slide 11 in the book here, and we see, note the crosshatch indicates a big space here, and what was not explicitly demonstrated in the Tanagawa experiment, but is implied, is that great diversity is achieved by putting different V regions next to the constant region. So here we see a bunch of V's, bunch of J's, a space here, and then a space to the constant region. And then in actual B cells, they're put together. V and J are next to each other, and they're now closer to the constant region. The J to C relationship is not a DNA rearrangement. That's a splice. You know about RNA splicing. Just because you have this, you've got RNA splicing. You still got RNA splicing. Yet don't confuse the DNA rearrangement with RNA splicing. Remember, virtually every eukaryotic gene does RNA splicing, but DNA rearrangement is only done in antigen receptor genes in immunoglobulins. So now, a lot of people were getting in the act. This is a pretty hot thing at that time, and people began to look at heavy chains now, a little more complex. Lee Hood, who has spent, I don't know if he's still active, but has been here at the University of Washington. At this time he was at Caltech. For more recent years he's been at the University of Washington. He was a leader in doing this, especially scaling up, getting massive amounts of sequencing done. So looking at heavy chains, Hood found that amino acids 1 to 101 were in a V segment, 107 to 123 in a J, and then 102 to 106 in yet another segment called D for diversity. Not surprisingly, this diversity segment ends up being the area of greatest diversity by comparing sequences, but also that we find out later is what's most closely associated with the antigen, where you're going to need the most diversity, right? And here is the genomic locus for heavy chains. We have these many V's, D's, J's, and C's. Note these big breaks here. And in the final B cell, these guys come together. Notice that this model requires that you lose the segments between the joints, between the V that's used and the D that's used, and between the D that's used and the J that's used. Okay. The J's downstream are not lost because that's a splicing event. And actually, talk about this later, the order, I gave that in reverse order. The DJ joining occurs first, and then V to DJ later. Okay, everybody still with me? So now we have 4 distinct segments. Diversity arises from the random use of one segment of each type. Essentially random. People have done things like trying to find usage, and it's close to random. But there's more diversity coming up. I feel when I present this, one of you has heard me present this kind of stuff in Bio 260, and sorry if I use the same lines, but I feel like a pitch man on late night TV. They say, you get this, you get this, you get this, and there's more. So there's this kind of diversity and this kind, but there's actually more. Create more diversity because a lot of diversity is needed. So here are some, this is kind of minutia, but there are 2 forms of light chain called kappa and lambda. They're enough different here, they call them different names. So C kappa or C lambda. In the mouse, almost all the light chains are kappa, very few are lambda. In humans, it's a little more equal. But here we have this kind of, this organization, and note, you know, this is kind of one of those slides to wow you. Don't worry about this too much with it, but there's multiple constant constant gene segments, and some of those are listed here. You see the general idea for the organization. By the way, again, this is called, the whole process is called gene rearrangement, and an individual V is properly a gene segment. Okay. And we see a lot of V kappas here in this organization, and many fewer lambdas in the mouse. You know, I've written down in the mouse only 5% of antibodies are lambda, only 3 functional V segments, and J's and C come in pairs. Okay. Humans are split a little more evenly. Humans have about 40% lambda. Okay. Now, so let's look at what kind of diversity we really have here. Let's look at this. So here are the number, this is in humans, and don't take these exact numbers. The truth is sometimes there's different haplotypes that might be a little bit different. Occasionally there's a mutation that's non-functional, but get the general idea. So in humans, number of, for heavy chains, about 45 V's, 23 D's, and 6 J's. So probability, multiply those together, make it 6,000 possible. For kappas, 41 V's, 5 J's, 205. For lambdas, 33 V's, 5 J's. So from these numbers, 2.3 million antibodies possible. Well, that's a lot, but still maybe not as much as we maybe could have or would like to have. What we observe that is happening. Okay. This gives you an idea. Because here, you read a heavy chain, any heavy chain can pair with any light chain, at least to a first approximation. Okay. So, there are these clusters of heavy chain gene segments. The D's can be read in multiple reading frames in human. So that's more diversity. So it's clear this is a major source of diversity, but there's still more. And this was found when they looked at the junctions, when they were able to do actual sequencing of junctions between V and D and J. And the next section talks about the mechanism of VDJ recombination. And as is common in molecular biology, I think you know this terminology, cis-acting sequences and trans-acting factors, right? Makes sense? Tell me if you'd like to say something about that. So for instance, for a promoter that governs expression of a gene, typically there are sequences right in front of it, upstream, and there are typically proteins that bind to those sequences. So we say those sequences are cis-acting because they can only act right there. Okay? The factors are trans-acting because they can be made elsewhere in the genome and then impact different places. Okay? So, like many other things in molecular biology, we find here that this event occurs because of cis-acting sequences and trans-acting factors. These cis-acting sequences are called recombination signal sequences, RSSs. We found that some of the enzymatic activities were specific for antigen receptor genes, for doing this, and some were common for general cellular maintenance. One of the things a cell has to do is DNA repair, and when there is a break, like from radiation, ligating DNA together. So there's a lot of mechanisms available for repair, for joining up DNA, and some of those genes and proteins that are involved in general repair, not surprisingly, have a role in gene rearrangement in immunoglobulin genes. So clearly this whole thing must be tightly controlled. Here are some of the proteins involved in VDJ recombination. RAG 1 and 2 are specific to these immune system cells. Recombination activating genes 1 and 2. They are close together. When I was learning some of this stuff, the point was made that they found this by transfection, and they were lucky that the two genes were close together in a genomic library, because if they weren't, they wouldn't have found them by that method. So RAG 1 and 2. Consequences of deficiency, severe combined immunodeficiency. Again, the relationship between actual disease, observed phenotype, and basic molecular biology. Of course, we do experiments. You guys are familiar with knockout experiments. Heard them, many of them discussed, but often clinically, people have observed something and then found the molecular basis later. Severe combined immunodeficiency. This enzyme called terminal transferase, TDT, adds non-templated nucleotides to the joints. Okay? The VD joint and the DJ joint. Non-templated, I think it's unique in that. Non-templated, just this adds on randomly, whatever ACTG. So what does that do? More diversity. Remember I promised you more? So there's the variation of the different segments, but there's even more diversity here. Okay. Here are some other non-lymphoid specific proteins that are involved, and these have roles in the non-homologous end joining pathway. So just the important thing is to know not what each of these does, but that there are lymphocyte-specific proteins, but also general DNA repair proteins. DNA ligation, some of these things here, usual DNA polymerases. I'm only going to mention the ataxic telangiectasia mutated protein. Has anybody heard of that disease? It's pretty obscure, but I worked in a lab where that was one of the main focuses for a long time, and don't ask me about what's going on now, but it's involved in signaling also when there's a break. People who have defects in that protein are very sensitive to radiation. So it was ultimately found that besides the recombination, let's think of the different mechanisms here, that the different segments, diversity of segments, which V with which D with which J, and then which heavy chain with which light chain, their diversity, and then talk about this a little further. We have this non-templated addition of nucleotides at the joints, and also sometimes there's chewing in or nibbling in at the joints. So together, this leads to really a lot of diversity, enough for you or a mouse to be able to fight anything. Okay, convinced? Okay, that's a lot of diversity. Amazing to think of the, all the steps and how all these things arose. And also, it's interesting to think about the common proteins and mechanisms used in general DNA repair and joining, and some things that are specific to lymphocytes. So here, I kind of glossed over this, but for each of these, there are people known with defects there, and they tend to lead to severe problems, including SCID, severe combined immunodeficiency. Combined means both the B cell arm and the T cell arm of the immune response. Okay, so now I'm going to go a little further here, thinking about steps here. So how does this happen? We talked about kind of the end result, but how does this happen? What is the order? So these, our knowledge of these rearrangements gives us another milestone in differentiation of B cells, going from the unrearranged to partial rearrangement to complete rearrangement. So in analyzing B cell development, which is something people make careers out of, the phenotypes, the mileposts one could look at include status of the DNA and also markers on the surface. Immunology is big on markers on the surface, as you know already. So first, early on in what are called pre-pro B cells, let me see what this is here. There is DJ joining. There's DJ joining in the bone marrow, and then later V to DJ occurs during what's called the pro-B stage. The rearranged heavy chain that's now formed pairs with what's called a surrogate light chain. Okay? So that's what's seen here. And so this is not the final light chain that's going to recognize antigen, but this is a way for the cell to say, everything's okay. Okay, we got heavy chain rearrangement down, we can move forward. And this allows the starting light chain rearrangement. Now, light chains are rearranged during a stage that's called a small pre-B stage. And then, if kappa works in mouse, that's great. If not, then they can try to rearrange lambda. When I say if something works, it means, I should have made that more clear on that. In the heavy chain in rearrangement, if, you notice I said there can be nibbling or addition of nucleotides, you might have thought, some of those are going to be non-functional, right? Because they're going to be out of frame. And in fact, some of those are. So there's a check to make sure things are okay. So when a real mature heavy chain can be made, it can come to the surface with a surrogate light chain. So here we now have a mature with B cell receptor with heavy chain and light chain. When a good IgM is made, it shuts off light chain rearrangement. So note the regulation to ensure things are okay, to get a good receptor. This works one allele at a time. Once one works, it stops. So people, of course, what do molecular biologists do? We look for sequences, recognition sequences, and the transacting factors. So these are what they found for the recognition, the recombination signal sequences, and it's a little bit unusual in that these sequences have a spacer that can be any sequence in between, and then defined sequences at either end. And this has, these RSSs have a 23 base spacer or a 12 base pair spacer. And the spacers are not conserved across different genes. And the same pattern is true on the recombination signal sequences for light and heavy chain. What happens, the cell brings together one RSS with a 12 base spacer and one with a 23 base spacer. This is the location of the RSS is here. Upstream and downstream, as you might expect. And we see here for heavy chain, notice the D segment has to have the RSSs upstream and downstream. Also note that because of this, because of needing to have one of each type, you can't have direct V to J joining in heavy chains. Okay, so here's a slide now. For light chain, germline, you see these RSSs upstream of these J's and downstream of these V's. Here, there is recombination between this V and this J. When that occurs, the J's that are upstream of this are lost. In some cases, they found they can isolate circles where they're lost. For heavy chain DNA, V cluster, D cluster, J cluster, first DJ rearrangement, and then V to D, V to DJ rearrangement. I guess I could have had these a little bigger for you. Okay? So that's the function of the RSS, the recombination signal sequences, the cis-acting sequences, and again, the transacting factors include the RAG proteins. So this has obviously been an area of intense study, and you don't need to get into detail of this stuff. This is what I told you don't have to read, but here we have, this is the intervening DNA, and the model is a loop with the proteins with RAG2 and RAG1 playing this role here. And again, not surprisingly, people have, here's this structure here, and even looked at the RAG1 and RAG2 proteins and these different domains that are important for them. So these next slides, you really don't, this is the stuff that I said don't worry too much about, but just know that there are a number of proteins necessary, some lymphoid specific, some general. RAG1 and RAG2 are lymphoid unique to lymphocytes. Other proteins are part of the non-homologous end joining pathway. Note that people with DNA repair diseases have immunoglobulin deficiencies. Not a surprise. They can't do normal DNA repair in response to insults like irradiation. They also have deficiencies in antibodies. mentioned that the transacting factors, RAG1 and RAG2, are close together in the genome. Their expression is developmentally regulated, only expressed in the early stages of B cell development and T cell development. This begs the question of why do we regulate the rearrangement of immunoglobulin gene segments in B cells but not in T cells? Why do we get the rearrangement of T cell receptor gene segments in T cells but not in B cells? And it's kind of an arm-waving thing, but there is some thought about changes in chromatin structure in so-called sterile transcripts, not coding for anything that happened before, as the area is opened up for attack by these recombination enzymes. I mentioned the anecdote about the RAGs being close together, hoping to us to understand their discovery. Okay, so I won't go in detail about what they mentioned about active chromatin. That's one of the sections I said to ignore. So I think I will stop at this point. It won't be too much of a spoiler to tell you that the story with the T cell receptor in T cell receptor is made up of two genes, two chains, alpha and beta, beta analogous to the heavy chain of immunoglobulins, alpha analogous to the light chain of immunoglobulins. Again, RAG, again, one has V, D, and J, one has just V and J, and also, again, a regulated, controlled program of gene rearrangement and checking to see, is this working, does this work, does this work? Okay? And we'll continue some of that in the next lecture, which will be the asynchronous lecture on Friday. I think I'll stop here. Again, note the value of these signposts or milestones in B cell development. Different stages can be characterized by what part of rearrangement has occurred, heavy D to J, then heavy V to DJ, when light chain rearrangement is seen, when there are proteins on the surface. Okay. So any questions about anything given today?